Lecture 43, Parasitic Neural Manipulation, When the Mind is Not Its Own. In the grand theatre of evolution, one of the most disquieting subplots is the story of parasites, organisms that do not merely feed upon their hosts, but alter their hosts' very behavior. Sometimes the changes are delivered as subtle nudges, other times they are grotesque rewrites of instinct and volition. This lecture explores the strange and often horrifying world of parasitic neural manipulation, where an external agent hijacks the nervous system of an animal and reprograms it to serve ends entirely foreign to the host's own evolutionary interests. This is not symbiosis. This is usurpation of the mind. Parasites capable of behavioral control span a broad swath of the tree of life: viruses, protozoa, fungi, worms, even other insects. Yet among these, there is a spectrum between indirect physiological subversion and direct behavioral control. Many parasites influence a host's body or health in ways that incidentally change behavior, for example, causing weakness or deformity that leads to easier predation. But only a rare few have evolved to directly and purposefully act on the host's nervous system, using molecular signals, anatomical invasion, or evolved mimicry of the host's own neuromodulators to induce specific behaviors beneficial to the parasite's life cycle. These are the neuroparasitic elites, the mind controllers, our focus here. To ground this discussion, let us begin with one of the most well-documented examples of mind control in nature, the lancet liver fluke, Dicrocelium dendriticum. This tiny flatworm has a complex three-host life cycle that takes it from land snails to ants and finally to grazing mammals like sheep or cattle. While inside its intermediate ant host, the fluke performs a kind of neurological prestidigitation. A few of the ingested parasite larvae migrate into the ant's nervous system, specifically to a cluster of nerves in the ant's head, the subesophageal ganglion. There, they secrete neuroactive compounds that hijack the ant's motor and sensory circuits. As evening falls, the infected ant feels an irresistible urge to abandon its normal routines and instead climbs up vegetation, often to the tip of a grass blade, and clamps down with its mandibles. All through the cool hours of dusk and dawn, the ant remains fixed in this unnatural position. This aberrant behavior drastically increases the likelihood that a grazing mammal will inadvertently ingest the ant along with the grass, delivering the parasite into its final host where it can mature and reproduce. After sunrise, as the day grows warmer, the spell is broken. The ant, if still alive and uneaten, unclenches its jaws and climbs back down to resume normal foraging behavior during the day. Remarkably, the parasite's manipulation operates on a daily cycle, switching on and off like clockwork. This circadian hijacking is exquisitely precise, reflecting a deep evolutionary entanglement between the parasite and the host's neurobiology. The fluke essentially presses a climb up and clamp button built into the ant's neural circuitry, but only at the time that maximizes the parasite's transmission to a grazing herbivore. By sparing the ant during the high-risk daylight hours, the parasite preserves its transport for future nights, an astonishing example of strategic, fine-tuned mind control. Now consider an even subtler puppeteer, the protozoan parasite Toxoplasma gondii. T. gondii's definitive host, where it reproduces sexually, is the cat family, but it can infect virtually any warm-blooded animal as an intermediate host. When Toxoplasma infects a rodent, say, a rat or a mouse, something happens that almost defies belief. The rodent's innate, hardwired fear of cat scent, specifically, cat urine odor, is not just dulled, but dramatically reversed. Instead of cowering or freezing when it smells a cat's territory, the infected rodent becomes curious, even attracted to the scent of the very predator it ought to avoid. It's as if a primal fear circuit in the rodent's brain has been unplugged and, in some cases, even rewired into a misguided approach behavior. Studies have shown that infected rodents explore novel areas more boldly, show less anxiety in open spaces, and specifically lose their aversion to feline odors. Remarkably, some experiments suggest they may even seek out places where cat urine is present. The result is a rodent that is far more likely to blunder into a cat's jaws, delivering toxoplasma back into a cat host to complete the parasite's life cycle. Even at the level of mate choice, hosts fight back. Females of some species prefer parasite-free males when choosing mates, as indicated by brighter plumage or vigorous courtship dances that parasitized males can't perform. By selecting healthier, non-parasitized mates, they reduce the risk of their offspring inheriting parasites or being exposed. This is part of what's known as the behavioral immune system in humans and other animals. We experience it often as disgust or wariness, the feelings of revulsion toward rotting food, foul smells, or obviously ill individuals can be seen as evolved behaviors to avoid sources of infection. In humans, the behavioral immune system can manifest in social ways. People may avoid or stigmatize those who appear sick or even who are just foreign, which, in ancestral environments, could correlate with carrying unfamiliar pathogens. While such reactions can be problematic in modern contexts, they likely have roots in disease avoidance. Now, although these defenses are not aimed at neuroparasites specifically, we didn't evolve specifically under pressure from zombie wasps. They reduce the opportunities for parasitic manipulation simply by reducing parasite transmission. If you don't get infected in the first place, you won't be controlled. But intriguingly, the behavioral immune system might sometimes overshoot, leading to pathologies of its own. Extreme or misfiring disgust and fear of contamination can result in conditions like obsessive-compulsive disorder or severe phobias. It's as though the mind, in guarding its integrity, can become overzealous and create new problems, a kind of autoimmune disease of the psyche. Another line of host defense is neural and cognitive flexibility. A host that can rapidly adapt its behavior, learn from new situations, and override its immediate impulses is much harder for a parasite to manipulate with a one-size-fits-all trigger. Parasites often exploit fixed action patterns or stereotypes, simple stimulus-response loops that are predictable. Insects, for example, often have very stereotyped behaviors. Ant goes up grass when temperature drops, cricket walks toward light reflection, etc. If the parasite can figure out the key stimulus or internal trigger, it can reliably induce the behavior. But imagine a more cognitively complex animal, say a crow or a pig. Such animals can learn from experience, and their behavior can be less rigid. If something compels them to do X and they start to do it but then realize it's harmful, they might stop. Or they may have more complex motivations that can counteract each other. Parasites would have a harder time orchestrating a precise outcome in such a case. It is perhaps no accident that many of the clearest examples of mind control come from insects and other small-brained creatures. That said, as toxoplasma shows, even mammals can be tampered with, but often in subtler ways, shifts in anxiety or activity levels rather than puppet-like control. This has led some researchers to propose an intriguing hypothesis, that the evolution of higher cognitive abilities and more flexible behavior in some lineages was partly driven by pressure to avoid being manipulated by parasites. In other words, just as predators might drive the evolution of speed or camouflage in prey, perhaps parasites, especially those that mess with behavior, could drive the evolution of more sophisticated brains and behaviors that are harder to predict or control. If a parasite's success relies on host behavioral predictability, then from the host's perspective, being unpredictable or having greater self-regulation could be advantageous. Think of it this way: a cricket doesn't know why it suddenly feels compelled to jump in water, it just follows the urge. A human, on the other hand, when feeling an odd impulse, can introspect, that's strange, why do I feel that way? Maybe I shouldn't do this. That very ability to sense an internal aberration and counteract it could be a defense that advanced brains have. This is highly speculative but thought-provoking. It redefines aspects of what we call executive function or self-control as not just tools for problem-solving in the environment, but also as a sort of metacognitive immune system. The brain's ability to say, this impulse might not be in my best interest, I choose not to act on it. We'll revisit this notion when talking about free will. But even in a simpler sense, a tendency for behavioral innovation and variability, not always doing the same thing in a given situation, could make a host a moving target for a parasite. In an evolutionary sense, hosts constantly changing the locks on their behavioral programs forces parasites to either become extraordinarily adaptable or give up. Many parasites seem to go for very basic, deeply conserved behaviors, like locomotion, feeding, mating, because those are ancient and shared across many individuals with little variation. Hosts in turn could evolve modifications or greater plasticity in those behaviors. There is another form of defense that is cultural rather than biological. In highly social and intelligent species, humans being the prime example, knowledge can be shared across individuals and generations about parasites and how to avoid them. Folklore, myths, and religions through the ages have warned of possession and mind-altering demons. While couched in supernatural terms, some of these cultural narratives reflect real dangers, like disease or parasite infection, translated into the language of the time. For example, stories of evil spirits that can invade and change a person might have encouraged behaviors that incidentally prevent parasite infection, such as avoiding certain areas, foods, or animals associated with those spirits. Some cultures developed rigorous hygiene practices, either as secular knowledge or as religious ritual, e.g., dietary restrictions, cleansing ceremonies, taboos around contact with the sick or the dead. These can dramatically reduce parasite transmission. Consider that in medieval times, people believed in demonic possession which, in a metaphorical sense, is not entirely unlike what rabies or toxoplasma or other neuroparasites do. Exorcism rituals or quarantine of the possessed might have occasionally saved communities from outbreaks of rabies by isolating those afflicted, even if they misunderstood the cause. The point is that culture itself can encode anti-parasite measures. Humans uniquely can identify a threat and disseminate a preventive strategy, even if mythologized. In the context of our lecture, one could say we have mimetic antibodies, shared ideas to guard against mind attacks. For instance, the fear of being hexed or poisoned might make one cautious about unknown influences. These aren't foolproof or always rational, but they show that the battle can occur in the collective mind as well. Yet, for every defense the host mounts, parasites often counter-adapt with new subterfuge. Some parasites actively suppress the host's immune responses to slip into the brain unopposed. For example, toxoplasma releases molecules that downregulate inflammation, like reducing certain cytokines, which both helps it survive and might contribute to the calmer behavior of the host. Since an inflamed brain tends to produce anxiety, by preventing inflammation, the parasite might be indirectly inducing a more serene, fearless state. Many parasites produce molecular mimics of host hormones or cytokines. Some schistosome worms produce peptides that look like the host's own opioid peptides, endorphins and enkephalins. These not only help the worm by modulating the host's immune cells, dampening pain and immune response, but also could affect the nervous system, since those opioids can cross into the brain and influence mood or stress. By mimicking the host's own biochemicals, parasites can fly under the radar, bypassing pattern recognition receptors that normally spot foreign molecules. It's the equivalent of a hacker using a trusted system process to carry out malicious activity, rather than a blatantly rogue code that would be flagged. Some parasites go even deeper, manipulating the host's gene expression. There is evidence that certain viruses and intracellular parasites can alter epigenetic marks, chemical modifications on the host DNA that control gene activity in neurons. For instance, rabies virus doesn't just kill neurons, it also interferes with neurotransmitter receptor expression and ion channel gene expression, possibly by introducing viral proteins into the nucleus that affect transcription. The result could be neurons that fire more readily in aggression circuits or that fail to respond to calming signals. Another example, some wasp-associated viruses, like polypnoviruses, act actively alter transcription in host caterpillar cells to disable the immune system and could plausibly alter neural cell function as a collateral effect. We are only scratching the surface of these molecular battles, but parasites essentially behave like biological hackers, constantly evolving new exploits to take over the host's operating system, the CNS, for their own benefit. Meanwhile, the hosts evolve patches to fix vulnerabilities, and so the cycle continues. This co-evolutionary struggle is an illustration of the Red Queen hypothesis. Both host and parasite must keep running, evolving, just to stay in place relative to each other. If the host evolves a new resistant behavior or detection method, the parasites that can circumvent or counteract that will have an advantage and spread, thereby pressuring the host to evolve further changes. In the context of neuroparasitism, this means the host's cognitive architecture, what it pays attention to, what it fears or likes, how flexible its instincts are, may be partly shaped by an evolutionary history of resisting mind control. It's a provocative way to think about why certain behaviors exist. Perhaps some of the quirks of animal, or human, psychology, like an innate fear of certain things, or a tendency for self-reflection, were influenced by the need to avoid being manipulated. For instance, an ant species heavily preyed on by a brain-controlling parasite might evolve either to recognize and remove early infected members, or individuals might evolve a tendency to avoid the situations that the parasite tries to exploit, maybe a genetically influenced aversion to climbing high at twilight, say. If that trait spread, the parasite would be in trouble unless it counter-evolved to push a different button. One fascinating, though speculative, possibility is that some hosts might evolve internal security systems, brain circuits that detect when one's own behavior deviates from the norm and then take corrective action. Imagine an animal that normally is shy of predators. If some internal or external influence suddenly made it bold toward a predator, perhaps a second-order mechanism could notice, something's off, I'm doing something dangerous I wouldn't normally do, and then suppress the impulse or even induce the animal to hide until the feeling passes. Sickness behavior in animals, the tendency to withdraw, lose appetite, and rest when ill, could in part serve this function. By becoming lethargic and reclusive when fighting an infection, a host might also inadvertently limit a parasite's ability to make it do crazy things. If a parasite wants you active and out getting eaten, but you feel too sick and just hunker down, the parasite's agenda is thwarted. It's like a safe mode where non-essential behaviors are turned off. Some researchers have indeed speculated that behaviors like depression or severe apathy during infection could be an adaptive response to avoid risky behavior that a parasite or pathogen might exploit. It's a bit of a stretch, but it speaks to the subtle back and forth at play. Stepping back, we see that the host-parasite battle does not merely play out in fevers and immune cells, it plays out in neurons, synapses, and behaviors. It has likely shaped cognition itself. It challenges the notion that brains evolved solely for solving ecological problems like finding food and mates. Perhaps an underappreciated driver of neural complexity in some cases was this invisible war of influence. Every sensory input, every decision-making rule, every emotional bias in an animal's brain could be seen as part of a tug-of-war between the host's evolutionary interests, survive and reproduce, and any manipulative parasite's interests, often, get eaten by something else, or aid the parasite's reproduction even at host's expense. Thus, what an animal perceives, or fails to perceive, what it learns easily or struggles to learn, and what it is motivated or hesitant to do, all might bear fingerprints of this ancient war. The host's perceptions and desires are its defense and also the parasite's entry points. In the final portion of this lecture, let's turn explicitly to philosophy and the broader implications of parasitic neural manipulation. The biological stories we've recounted, from ants clamping to grasshoppers drowning to rats courting danger, are remarkable on their own terms. But they also prompt us to ask, what do these scenarios tell us about ourselves? About concepts like free will, agency, and selfhood? When we confront the idea that a parasite can change who an ant is, or at least what it does, we inevitably reflect on how much of our behavior is truly under our own control. Start with the deceptively simple question we raised. What is a self? Neuroscience often describes the self not as a single unified entity, but as an emergent property of various brain processes, a construct that integrates our perceptions, memories, and goals into a narrative or coherent point of view. It's not a hard-coded thing, it's continuously generated and updated. In many spiritual traditions, like Buddhism, similarly, the self is considered an illusion, or at least not a fixed thing, just a collection of momentary components, sensations, thoughts, reactions, that we mistakenly perceive as one enduring identity. Modern neuroscience and ancient thought converge on the idea that the self is constructed and somewhat fragile. Now, if the self is a pattern that the brain produces, rather than an inviolable essence, then it stands to reason that it's possible to perturb that pattern and shift the self. A parasite doesn't need to mind control, in a sci-fi sense of implanting new thoughts. It can just nudge some of the parameters of the system. A little extra dopamine here, a little less GABA there, a slight inflammation in this region, and suddenly the emergent psychological state, the thing we call self or mind, is altered. The parasite doesn't destroy the brain or even necessarily damage it in any obvious way. It just tunes it to a different station. The ant's neurons, the rat's neurons, are still doing what neurons do, but now the frequency and context are off, and the behavior that emerges is maligned to the host's own survival interests. This recognition hits close to home when we think about more familiar influences on our behavior. For instance, our gut microbiome, the teeming community of microbes in our intestines, is known to produce neurotransmitters and modulate our physiology. Certain gut bacteria produce serotonin. A majority of the body's serotonin is actually in the gut. Others produce GABA, dopamine precursors, or short-chain fatty acids that affect brain function via the vagus nerve or immune system. While these microbes are often benign or symbiotic rather than parasites, their influence blurs the line of identity. If a bacterium in my gut produces a compound that makes me slightly more anxious or bold, is that my anxiety or boldness, or the bacterium's doing? It's a meaningless distinction in one sense. It's all chemistry in my body, but it challenges how we assign agency. Consider cases where gut microbiota have been implicated in mood disorders or even social behavior in animal studies. Swap the gut bacteria of a shy mouse with those from an outgoing mouse, and the shy mouse may become more gregarious. That sounds eerily similar to parasite experiments. We don't typically label gut flora as mind-controlling parasites, but from a systems perspective, they kind of are affecting our brain from the inside. They just happen to usually align with our well-being because a healthy, socially functioning host is good for them too. But the conceptual leap is that the puppet strings of behavior can originate from within as well as from without. If we consider a microbe that lives in us and influences us, is it part of us or an intruder? Does it matter for the notion of self? A molecule is a molecule whether it came from your own neuron or a bacterium in your colon or a parasite in your blood. If it triggers a receptor in your brain and changes your mood or behavior, it has effectively influenced you. The tongue-in-cheek question arises, is a molecule produced by a microbe in your gut any more self than a molecule injected by a wasp stinger? We are inclined to differentiate them. One is part of normal physiology, one is an external attack, but perhaps from the perspective of our neurons, they are just signals to interpret. Extending this line of thought, we confront not just biological parasites but mimetic parasites. The evolutionary biologist Richard Dawkins coined the term meme for ideas or cultural units that spread and evolve somewhat like genes. Some ideas can be thought of as parasitic in that they propagate for their own sake, not necessarily for the host's benefit. For example, an urban legend or conspiracy theory that causes people to take dangerous actions can be seen as a mind virus. It spreads by altering host behavior, telling others, acting on the belief, often to the detriment of the host or society, but beneficial to the meme's spread. Social media algorithms today inadvertently act like incubators for such memes, selecting for content that grabs attention and triggers emotion, fear, anger, tribalism, because that content gets more engagement, and thus spreads further. A meme that says, "Spread me or you will suffer," think of chain letters or certain extremist ideologies, has an intrinsic advantage in the marketplace of ideas, much like a parasite that compels the host to help transmit it. We can draw a parallel. Toxoplasma makes a rat approach a cat, effectively a suicide mission that helps the parasite. Similarly, a dangerous meme might make a person perform actions that could be self-destructive or against their own interests, in order to propagate the meme. Consider cults where members sacrifice their well-being for the group's belief, or online challenges that risk health. It might sound abstract, but it's increasingly salient in the age of misinformation, ideas that hijack our neural reward and social circuits, by giving us outrage to bond over, enemies to hate, or a sense of belonging to a crusade, can cause us to act in ways that benefit the idea's spread even if it harms us or others. While not a biological parasite, it's an information parasite that uses the same pathways of influence, fear, reward, social identity that biological ones do. So, when we contemplate free will in light of all this, it stops looking like a binary concept, you have it or you don't, and more like a gradient or spectrum of susceptibility. Perhaps we can define autonomy as the capacity to resist being manipulated, whether by a parasite, a drug, an impulse, or a meme. The more an organism can maintain alignment between its actions and its own survival, reproductive interests, or personally chosen goals, the more free will it effectively has in an evolutionary sense. In contrast, if an organism's behavior is easily hijacked by others, viruses, parasites, or even social pressures, its agency is diminished. By this definition, a lot of human behavior might fall somewhere in the middle. We are more autonomous than the ant with the lancet fluke, but are we entirely self-directed? Advertisers, political propagandists, and persuaders of all kinds are constantly trying to parasitize our decision-making for their ends, money, power, etc. Every time we buy something because an ad manipulated our emotions, or share a piece of clickbait without thinking, one could argue we've been slightly parasitized by an idea planted in us. This isn't to equate advertising with a hairworm, but the structure of the phenomenon has similarities. Autonomy, then, could be reframed as a sort of resistance trait, perhaps even evolving like one. Some individuals, or species, might be naturally less susceptible to certain manipulations. For example, not all rats infected with toxoplasma completely lose fear. There are differences. Similarly, not all people are equally swayed by misinformation or peer pressure. Some have a more skeptical or independent cast of mind, whether through genes, upbringing, or practice. In evolutionary terms, when parasites are common, those who resist manipulation better should have a survival advantage. In human terms, perhaps those who can critically evaluate information and regulate their emotions avoid some modern mind traps. Of course, no one is immune to everything. But thinking of free will as the margin we have to assert our own purposes in the face of myriad internal and external influences is a useful perspective. It's not an on-off switch, it's more like a muscle that can be strengthened or a shield that can be fortified. This perspective has potent ethical implications. We usually think of ourselves as responsible for our actions because we assume we are the authors of those actions. But if an action is heavily influenced by a parasite or a pathology, we tend to reduce blame. The individual wasn't themselves. For a clear-cut example, consider a dog with rabies that bites someone. We don't think of the dog as evil. We understand the virus made it hyper-aggressive and not fully in control. In humans, too, there are cases that challenge our notions of responsibility. There's the famous case of a man who developed pedophilic impulses because of a growing brain tumor. Once the tumor was removed, the impulses went away. When the tumor recurred, so did the behavior. This was a purely internal parasite, of sorts, a lesion altering his drives. The legal system grapples with such issues via the insanity defense or diminished capacity arguments. If someone commits a crime while in the throes of a parasitic brain infection, say, neurocysticicosis causing psychosis, or under the influence of a mind-altering substance they were unwittingly exposed to what extent are they accountable? As we learn more about how subtle and not so subtle brain modulation can change behavior, the grounds for moral judgment get complicated. If free will is indeed an achievement rather than a baseline state, then some individuals might have compromised capacity for it due to factors beyond their control, genetics, infection, environment. This doesn't mean we abandon the idea of responsibility. Society couldn't function if we did, but it does mean we might move toward a more nuanced understanding. We already intuitively do this. A person with severe mental illness may be treated with compassion and therapy rather than punishment if their actions stemmed from delusions. Perhaps one day we identify, say, a parasite or microbiome imbalance that contributes to impulsive aggression in a person, and treating that could mitigate the behavior. The more we see how mechanistically fragile the scaffolding of our thoughts and decisions is, the more we might approach issues of behavior with a medical lens rather than a purely moralistic one. Even beyond pathology, consider the everyday fact that many of our fundamental drives can be manipulated by external chemicals. We intentionally do this through psychopharmacology, caffeine to stay alert, antidepressants to lift mood, beta blockers to quell anxiety, testosterone or estrogen treatments that can affect aggression or mood, etc. We have whole industries and institutions that, in effect, hijack brain chemistry for desired outcomes. Sometimes the outcome is to benefit the individual, as in medicine, but sometimes it's to benefit someone else. Think of the use of sedatives or even propaganda, which is a psychological drug of information, to control populations. In dystopian fiction like Huxley's Brave New World, people are controlled by a pleasure drug, soma, that keeps them docile. That's basically a parasitic strategy, albeit implemented by a government rather than a microbe. One could argue that the line between parasite and exploiter blurs when you consider things like drug dealers who get people addicted for profit. The drug is the mechanism, but the end result is the person's behavior now serves the dealer, keep buying the drug, at great cost to themselves. It's a harsh analogy, but structurally it's similar. The person's natural reward circuits are subverted to prioritize something that isn't in their genetic interest, spending resources on a substance rather than on survival or reproduction. In a less nefarious sense, even the comfortable modern life with abundant sugary, fatty foods and digital entertainment is hacking our ancestral neural pathways of reward, sometimes leading to maladaptive outcomes like overeating or screen addiction. Those aren't parasites, but they demonstrate how our brains can be gamed by stimuli that exploit them. So where does all this leave us, as thinking beings reflecting on these marvels and terrors of nature? Perhaps the most important lesson from the study of parasitic manipulation is that autonomy requires vigilance. We can't take for granted that our thoughts and desires are entirely our own or entirely in our best interest. Autonomy isn't the passive state of being in control, it's an active process of monitoring and checking influences. In biological hosts, the ones that fare best are not necessarily those with impenetrable minds. There may be no such thing, but those who can detect when something is off. We see glimmers of this in nature, an infected ant's nestmates detecting its aberrant behavior, or possibly an animal isolating itself when feeling sick. For us, it might be more introspective, the capability to notice, I'm feeling an oddly strong urge to do X, Y. Is that really me or something acting on me? This kind of self-reflection can be seen as a mental immune response. Just as your body's immune system flags foreign invaders, maybe part of what we call consciousness or self-awareness is about flagging foreign mental invaders, be they literal parasites, drugs, manipulative ideas, or even intense emotions. Strengthening that metacognition, being able to step outside one's immediate experience and evaluate it, could be one of our best defenses against both organic and synthetic manipulators of the mind. Interestingly, this idea resonates with certain spiritual and philosophical traditions. In Buddhism, for example, Practitioners cultivate mindfulness to observe their thoughts and feelings as transient events, not inherently self. One learns to watch cravings, aversions, and emotions arise and pass without automatically acting on them. In doing so, one gains a measure of freedom from them. If you can identify a desire as not truly you but as a phenomena occurring in the mind, you have the choice to let it go. In our context, that's the ultimate anti-parasite strategy, to be aware enough to say, I see I am being nudged in a certain direction, I will decline to follow that nudge. Whether the nudge is from a parasite's molecule or one's own misfiring circuitry or an external social pressure, the effect is the same. Self-awareness, then, is more than a philosophical curiosity, it's a weapon forged by evolution, or by personal cultivation, to maintain our integrity. Autonomy is not total independence, we are never free of all influence, but it is the capacity to recognize influence and choose how to respond. Finally, a rather counterintuitive silver lining emerges from studying these dark manipulations. Parasites, in their diabolical genius, can become teachers. They reveal by contrast and by exaggeration aspects of neurobiology that we might otherwise miss. Each parasite that manipulates a host is conducting an experiment, honed by time, on how to tweak a brain. By studying how they do it, we learn about the circuitry and chemistry of behavior in unprecedented ways. Consider that Ampulex wasp again. Its venom identified that blocking octopamine in specific neurons induces lethargy without motor impairment. This hints at where motivation lives in the insect brain. We've isolated compounds from parasite venoms and secretions that could become drugs or research tools. For instance, if a fungus produces an amphetamine analog to make cicadas hyperactive, that's a clue about insect, and perhaps even human, neurochemistry of activity and appetite. Indeed, we've found that Massospora fungi in cicadas produce cathinone, an amphetamine-like stimulant, and sometimes psilocybin, a psychoactive compound. These chemicals keep the infected cicadas buzzing with energy and sexual desire even as their bodies fall apart. Such findings not only confirm the mechanism of manipulation, the fungus is essentially drugging its host, but also open questions about why those particular substances and what else might they affect. In a broader sense, parasites show us the possible. An ant's mandibles can be made to clamp indefinitely, a mouse's ingrained fear can be erased, a fish's navigation can be hijacked. These give us insight into those behaviors in the normal state. They also inspire technologies. After all, human neuromodulation via deep brain stimulation or pharmacology is often crude compared to what parasites achieve. Can we learn to be as targeted? Could we treat disorders of the brain by mimicking parasites in benign ways, for example, using a viral vector to deliver genes that elevate dopamine in Parkinson's patients, which is not unlike toxoplasma elevating dopamine in a rat? It's a bit poetic that what we consider evil in nature might inform healing. Already some parasite-derived molecules are being studied for therapeutic use, such as immunosuppressants from worms to treat autoimmune diseases, or venoms for pain relief. It's not far-fetched that a wasp venom component could one day inform a psychiatric medication that dampens pathological fear or excessive impulsivity by acting on the same pathways the wasp does. As we conclude, we can't help but appreciate that the parasite is not just an invader, but a revealer. It reveals the contour lines of autonomy by pushing a behavior to its extreme. It shows us where the fault lines in the neural system lie. It exposes the soft underbelly of what we might romantically call the soul, demonstrating how things like desire, aversion, and intention can be puppeteered. It forces us to recognize that the machinery of decision and action is delicate and can be co-opted. But in doing so, it also highlights what is remarkable about a healthy, uninfected mind, the ability to choose, to have preferences aligned with one's own survival and well-being, to have agency. When those things are taken away, we see their value starkly. The mind, as these examples show, is not a fortress with impenetrable walls guarding an enlightened self. It's more like a landscape open to the elements, fertile but also vulnerable to weeds and floods and invaders. Under normal circumstances, we cultivate it and maintain its boundaries, but parasites show that under certain conditions, those boundaries can be trespassed. By studying these alien invaders of the mind, perhaps we gain a greater appreciation for the normal state of having our mind as our own. And we gain humility, realizing that the sense of self, of control, that we take for granted rests on biological processes that can be subtly subverted. In understanding this, we can strive both to protect that autonomy through scientific knowledge, through personal mindfulness, and to use this knowledge to better society, for instance, recognizing and resisting manipulative forces, be they infectious or informational. Parasitic neural manipulation is one of nature's darkest marvels. Yet in grappling with it, we shine light on the very essence of what it means to think, to choose, and to be oneself. What underlies this sinister change? At the neural level, toxoplasma installs itself within the host's brain, often forming microscopic cysts, particularly in regions like the amygdala, which processes fear and emotion, and other limbic areas. From these sites, the parasite releases compounds that interfere with the host's neurochemistry. One clue emerged when researchers discovered that T. gondii can produce an enzyme analogous to the host's own tyrosine hydroxylase, the enzyme that catalyzes the production of dopamine. In effect, the parasite may be chemically boosting dopamine levels in the rodent's brain. Dopamine is a neurotransmitter deeply involved in motivation and reward, and an excess could distort the animal's responses to stimuli. By ramping up dopamine, or otherwise modulating related signaling pathways, toxoplasma might dull fear responses and enhance exploratory, even risky, behavior. There is also evidence of localized inflammation and disruption of normal signaling in the olfactory pathways and amygdala, which could flip the innate fear response. The rodent's nose still detects the cat odor, but the emotional tag attached to that scent changes from terror to interest or indifference. In essence, the parasite performs a neurochemical switcheroo, recoding a predator odor from warning, cat, to something more like, what's that? This is not a mere side effect of illness, it appears to be a targeted reprogramming of the host's survival instincts. This phenomenon isn't limited to rodents. There is accumulating, if controversial, evidence that toxoplasma infection may subtly influence human behavior as well. Humans are not part of the parasite's intended life cycle, of course, but T. gondii can infect us incidentally, often via contaminated soil, water, or undercooked meat, or through cat litter. Intriguingly, large epidemiological studies have found correlations between latent toxoplasma infection in people and certain personality and behavioral traits. Infected individuals have, on average, shown higher risk, taking propensity, reaction time deficits, and in some studies even a slightly increased likelihood of car accidents. Some correlations have been noted between toxoplasma exposure and psychiatric conditions such as schizophrenia or intermittent explosive disorder. Causality is extremely difficult to establish in humans, and we must be cautious not to overstate these findings, but the mere possibility that a single-celled parasite could tweak the psychology of a primate is startling. It forces us to wonder, to what extent might some decisions or preferences we consider ours be nudged by hidden biochemical puppeteers? We will return to these musings on agency and free will, but first we must travel further into the menagerie of mind-controlling creatures. From here, the examples only grow more macabre and varied across the animal kingdom. Consider the hairworm, a long, thread-like parasitic worm, phylum Nematomorpha, that famously turns its insect hosts into unwitting swimmers. One species, Spinochordotes telinii, infects grasshoppers and crickets during its larval phase. The worm grows undetected inside the host's body cavity, sometimes reaching astonishing lengths, several times the host's body length, coiled within. When the hairworm is fully developed and ready to leave the host, it faces a challenge. Its next life stage is aquatic, but its insect host lives on land. Somehow, the worm solves this by brainwashing the insect into suicide by water. An infected grasshopper, at the behest of the parasite, experiences a compelling drive to seek water at night. A cricket that would normally avoid pools and streams will become restless, then walk or jump straight into a pond or puddle. In these unnatural waters, the insect often drowns, a death sentence for the host, but exactly what the parasite needs. Upon contact with water, the mature hairworm wriggles out of the dying insect's body and swims away to find a mate, complete its reproductive cycle, and release new larvae. The mechanism of this suicidal behavior has been the subject of intense research, and it highlights how sophisticated parasite manipulation can be. Hairworms do not appear to physically bore into the insect's brain, rather, they release molecules that diffuse into the host's nervous system and interfere with its programming. Researchers have identified proteins produced by hairworms that mimic some of the host's own neural signaling molecules. For instance, some hairworms produce analogues of WNT proteins, a family of signaling molecules involved in neural development and function. In a grasshopper's body, these parasite-derived WNT-like proteins can cross into the insect's central nervous system and potentially alter gene expression or synaptic connections. Essentially, the parasite hijacks the insect's neurodevelopmental pathways, possibly even rewiring certain circuits or eroding the insect's normal aversion to water. Other studies have found changes in neurotransmitter levels in infected insects. Serotonin and other monoamines may be altered, which could disrupt the insect's locomotion and perception. The result is a kind of delirium, the host's navigation and basic instincts malfunction. The grasshopper might perceive light reflecting off water as something to move toward, mistaking it for safe terrain or moonlit sky. Or it may lose the motor inhibition that normally prevents it from entering water. In effect, the parasite converts the insect's built-in movement routines into a death march. In a particularly elegant twist, if the grasshopper doesn't immediately drown and the hairworm emerges successfully, some host insects can actually recover partially and crawl back out of the water after the parasite has left. Like a puppet with its strings cut, the surviving insect may shakily return to something like normalcy within a day, suggesting that the parasite's influences, however devastating, were reversible and precisely timed to the worm's exit. Here we see parasitic mind control at its bleakest, a creature compelled to end its life in service of another's life cycle, a suicide that is not a suicide at all, but an act of biological coercion. If the hairworm is a subtle chemist, the jewel wasp, Ampulex compressor, is a neurosurgeon. This emerald green wasp is a parasite of cockroaches, and it has evolved a refined two-step method to effectively zombify a much larger host. When a female jewel wasp is ready to reproduce, she seeks out an American cockroach, stalking and pouncing on it like a tiny predator. In a burst of precision, she delivers 2 stings with her ovipositor, a stinger-like organ. The first sting goes into the roach's thorax, targeting a cluster of nerves that control the front legs. This venom injection immediately paralyzes the roach's front legs, preventing it from escaping or fighting effectively. But this is just to subdue the victim momentarily. The wasp's second sting is the truly surgical strike. She inserts her stinger directly into the cockroach's head, penetrating the protective exoskeleton and navigating toward specific areas of the roach's brain. In an almost unbelievable act of anatomical precision, the wasp injects venom into the region of the cockroach brain known as the subesophageal ganglion, essentially knocking out the circuits that initiate the escape reflex and locomotor drive. What happens next is extraordinary. The cockroach, a formidable and swift insect under normal circumstances, is not killed by the stings. In fact, it remains alive and able to move. But it no longer has any will to do so. The roach enters a state of profound lethargy known as hypokinesia. It will not run away or struggle, it simply stays where it is put, grooming itself intermittently and showing no initiative. The wasp, having effectively hacked the roach's decision-making center, then does something straight out of a horror film. She grabs the roach's antenna as if it were a leash and leads the zombified insect, which now obediently follows, into a burrow. There the wasp lays a single egg on the roach's underside and entombs the living but docile host in the burrow with some pebbles. Over the next week, the wasp's larva hatches and begins to feed on the roach, slowly devouring it from the inside while the roach, still alive, offers no resistance. Eventually the larva pupates and emerges as a new adult wasp, leaving behind nothing but the hollowed-out shell of a cockroach that never tried to escape its fate. What chemical tricks enable this level of control? The jewel wasp's venom is a complex cocktail of neuropharmacological agents evolved over millennia to incapacitate the roach without killing it. Analysis of the venom has revealed at least two notable components. One is a toxin dubbed AMPUTX, which blocks certain voltage-gated potassium channels in the cockroach's neurons. By blocking these channels, the venom likely prevents normal neuronal firing in specific circuits, effectively silencing those neurons. Another component selectively interferes with the cockroach's octopamine neurotransmitter system. Octopamine in insects serves a role analogous to norepinephrine adrenaline in mammals. It's central to arousal, fight-or-flight responses, and motivation. By inhibiting octopaminergic signaling, the wasp's venom essentially turns down the volume on the roach's arousal and motor circuits. The result is that the cockroach can move if physically pushed or prodded, its muscles and basic motor patterns still function, but it has lost the neural spark that would initiate movement on its own. It's as if the wasp pharmacologically lobotomizes the roach's will to move. What human anesthesiologists and neurologists attempt with precise doses of drugs in a clinical setting, this tiny wasp accomplishes with an instinctive sting guided by evolutionary trial and error. This is not mind control as metaphor, it is literal, biochemical control of one creature's behavior by another, pinpointed to specific neural substrates. The jewel wasp's larvae benefit from having fresh, paralyzed meat to consume, rather than a dead and decaying carcass, and so the wasp's venom has evolved to keep the prey alive yet utterly compliant. The level of specialization, venom that blocks exactly those signals needed for escape, without stopping the heart or killing the host, bespeaks an almost unimaginable evolutionary fine-tuning. Millions of years of natural selection have crafted a substance that makes a cockroach into a living pantry item and bodyguard for the wasp's offspring. Not all parasites achieve their manipulation through direct injection into the brain or by residing in the head. Some can control behavior by more insidious routes. In the marine environment, for example, consider the case of the tongue-eating louse, Cymothoa exigua. This creature is not a worm or a protozoan, but an isopod crustacean, essentially a parasitic relative of roly-poly woodlice. The tongue-eating louse targets fish. It enters through the fish's gills as a juvenile and then attaches itself to the base of the fish's tongue, where it proceeds to drink the tongue's blood supply. Over time, the fish's tongue withers from lack of blood, eventually dying and disintegrating. The parasite then firmly anchors itself in place of the old tongue, effectively becoming the fish's new tongue. The fish can use it to manipulate food and swallow as if it were a normal tongue, and the parasite gets a steady diet of the fish's blood or mucus. Now, one might argue this is more an example of parasitic replacement therapy than mind control. The louse isn't overtly directing the fish's behavior towards some new goal like predation or suicide. Yet, the integration is so seamless that the fish's nervous system now treats the parasite as a part of its own body. The louse has to coordinate with the fish's mouth movements, likely providing sensory feedback so that the fish can still eat. The fish, for its part, learns to operate with an alien organ. This involuntary prosthesis affects the fish's behavior and feeding patterns. Infected fish may have to compensate for the parasite's presence, perhaps taking different types of bites or feeding more frequently to get enough food for both host and parasite. It is a kind of occupation of the body that verges on the behavioral, because the fish's brain must now incorporate signals from and to this parasite. It raises the peculiar question of identity. Where does the fish end and the parasite begin, if the parasite functions as a piece of the fish's anatomy? It's not neural manipulation in the classic sense, but it shows how a parasite can become entwined with a host's neural and muscular systems to the point that the host's behavior changes to accommodate what is essentially a new part of its body. If a fish's tongue is no longer its own but an agent with a different genome, the fish's actions entirely its own. The tongue-eating louse, like many parasites, blurs the boundary between self and other in a very literal way. This theme, the blurring of agency, runs through all these examples. We confront a philosophical question: if a parasite can so completely and precisely alter a host's behavior that the host effectively serves the parasite's interests, is the host still itself? Is the ant atop the grass blade at dawn exercising any meaningful volition? The ant's neurons fire, its legs move, its mandibles lock, but the why of those actions lies in the fluke's life cycle, not the ant's. The cockroach walks under the wasp's guidance, and its decision not to flee isn't a decision at all, it's a chemically induced omission. At a certain point of complete control, the host becomes a vehicle, a puppet. And yet, the host is not dead, its brain is still the instrument playing the notes, just not its own composition. The concept of agency becomes fuzzy. There is no easy line where we can say the host's mind ends and the parasite's will begins, because in a sense the parasite's will is being executed through the machinery of the host's mind. Biology thus confronts us with scenarios that were once only imagined in science fiction or horror tales, identities layered on identities, one creature reaching into another's very nervous system to steer it, like an unseen rider on a horse. Before we spiral too deep into philosophy, let's broaden our scope and look at just how many ways parasites have evolved to influence hosts. The diversity of strategies is astonishing, spanning subtle biochemistry to outright anatomical overhaul. Some parasites achieve their ends not by directly seizing the neural helm, but by other forms of manipulation that nonetheless alter behavior. For instance, not all parasites that change host behavior do so with precise molecular tricks, some do it more bluntly. A trematode parasite called Ribeiroia infects developing frog tadpoles and interferes with their limb development, causing the frogs to grow grotesque extra legs or malformed limbs. These unfortunate frogs, once they metamorphose, can barely hop to escape predators. A bird, the parasite's next host, can easily catch the stumbling frog, allowing Ribeiroia to continue its life cycle in the bird's gut. This is a form of morphological manipulation with behavioral consequences. By physically hampering the host, the parasite ensures the host becomes easy prey. It's not neural manipulation per se, the parasite isn't fiddling with neurotransmitters in the frog's brain, but the end result is a behavior, or lack of behavior, inability to escape, that serves the parasite. This example underscores an important point: parasites can gain control over host outcomes in multiple ways, and the more primitive methods, like causing disease, pain, or debilitation that incidentally leads to behavior change, likely set the stage for more direct neural methods to evolve. Among the most sophisticated are those that act on the nervous system proper, and it's those we continue to focus on. Let's return to the realm of direct neural hijacking and examine how these parasites achieve such feats on a molecular level. Many use molecular decoys or mimics, chemical keys that fit into the host's neural locks. Earlier we noted the example of the hairworm secreting WNT family proteins in the grasshopper's brain. WNT proteins are normally crucial in the host for cell signaling during development and for synaptic plasticity. By flooding the host brain with a WNT mimic, the parasite may trigger gene expression programs or growth pathways that are out of place in an adult insect, possibly leading to a reorganization of neuronal connections or a breakdown of certain neural circuits, for instance, those that keep the insect fearful of water. It's like installing a new software patch into the insect's brain that changes how it responds to environmental cues. Similarly, other parasites produce compounds that mimic neurotransmitters or hormones. Toxoplasma, as mentioned, might be boosting dopamine levels or otherwise tampering with neurochemical signaling. Some parasitic wasps inject not only venom but also symbiotic viruses or virus-like particles that express genes in the host's nervous system. In the case of the wasp Dinocampus coccinelli, which turns ladybugs into twitching bodyguards for its cocoon, researchers found that an RNA virus carried by the wasp proliferates in the ladybug's nervous system. As the wasp larva is about to pupate outside the ladybug's body, the virus infection in the ladybug's neurons causes a kind of partial paralysis. The ladybug stands guard, unable to walk away, but still able to lash out and twitch to scare off predators that approach the wasp's cocoon. In some cases the ladybug even recovers after the wasp emerges, implying that the virus's effect was transient. Here, the parasite, the wasp, is effectively using a bioweapon, a virus, to manipulate another host's behavior. It's a multi-layered parasite manipulation, wasp plus virus working in concert to subdue the host's mind. Then there are the direct injectors like the jewel wasp we discussed. The jewel wasp's venom contains a cocktail of neurotoxins, neuromodulators, and enzyme inhibitors. We already highlighted components that block ion channels and octopamine. The net effect of this venom is akin to creating a targeted lesion in the cockroach's motivational circuitry, not physically but chemically. The roach's brain cells aren't destroyed, rather, their communication is jammed or muted in specific ways. The specificity is stunning. The roach loses the drive to initiate escape, but it remains capable of basic movement and metabolism. It's as if one could remove an animal's fear or free will with a syringe while leaving everything else intact. Other parasitoid wasps have similarly precise venoms. For example, some target spider hosts. One wasp that parasitizes orb-weaver spiders, such as Hymenopomyces argyraphiga, injects a chemical that causes the spider to abandon its typical web-building pattern and instead weave a very abnormal web, a special silk structure that happens to be perfectly suited for supporting the wasp's cocoon. The spider effectively becomes an unwitting architect, constructing its parasite's cradle, and once the structure is complete, the wasp larva kills the spider. The molecules in such venoms can include neuromodulators that interfere with the spider's web-building behavioral program, possibly by altering serotonin or other neurotransmitters in the spider's brain that govern web-building cycles. Interestingly, some parasites manipulate behavior indirectly by targeting the host's endocrine system rather than the neural circuits directly. The nervous and endocrine, hormonal, systems are deeply intertwined. Hormones can alter neural activity and vice versa. Certain parasitic nematodes in fish, for instance, secrete molecules that mimic or disrupt the host's hormones like cortisol or other stress-related hormones. One example, some worm parasites release analogues of corticotropin-releasing hormone, CRH, or other peptides that modulate the stress axis in the host. If a fish's stress response is dampened, imagine a parasite essentially administering a chemical that calms the fish or reduces its wariness. The fish might become sluggish and less responsive to predators. In other situations, a parasite might tweak hormones to make a host more risk-prone or agitated. Depending on what suits the parasite's life cycle, the host might either exhibit lethargy, making it easier for a predator to catch it, or for parasites to not be dislodged, or exhibit unusual boldness and activity, making it wander into danger. A dramatic case of endocrine-based manipulation is found in crustaceans infected by parasitic barnacles of the order Rhizocephala, like Sacculina. Sacculina barnacles inject themselves into crabs and grow inside their bodies, eventually castrating the crab and infiltrating its nervous system and hepatopancreas. These parasitic barnacles secrete compounds that radically alter the crab's hormonal profile. Male crabs infected by Sacculina stop producing normal levels of male hormones and often develop feminized features. Behaviorally, the male crab begins to act like a female carrying eggs. The crab ceases its typical mating behaviors and instead performs maternal care behaviors. It will aerate and tend the parasite's external brood pouch, which protrudes where the crab's own eggs would be incubated on a female. Essentially, Sacculina chemically castrates the crab and hijacks its reproductive instincts. The crab, in a sense, believes it is caring for its own offspring, when in fact it is nurturing the barnacle's larvae. This is a kind of deep identity theft. By altering hormone levels, like androgen and molting hormones, and possibly neuronal receptors, the parasite induces nurturing behavior in a host that normally would never show it. The neural circuits for maternal care are present in crabs, females use them, and Saculina simply replays that program in the wrong context, an infected male or even an immature female, all to protect and disperse the parasite's brood. It's not hard to see how close this comes to a literal loss of self for the crab. Its entire reproductive agenda has been overwritten. Even parasites that are themselves tiny microbes can exert endocrine-like manipulation. Plasmodium, the parasite that causes malaria, doesn't only affect humans, some species infect birds, reptiles, and even manipulate insect vectors. Studies on malarial parasites in mosquitoes, the vectors that carry Plasmodium, show that the parasite can alter the mosquito's feeding behavior depending on its own developmental stage. While the parasite is still developing inside the mosquito, not yet ready to infect a new host, it produces signals that make the mosquito less likely to bite and more likely to seek sugar, nectar, presumably to keep the mosquito, and parasite, safe and well-fed. Once the parasite's infectious stages, sporozoites, are ready in the mosquito's salivary glands, Plasmodium then causes changes that increase the mosquito's attraction to hosts and its propensity to blood feed aggressively. Essentially, the parasite times the mosquito's hunger for blood to match the parasite's readiness to spread, using chemical messengers that affect the mosquito's sensory preferences and endocrine signaling for hunger. This shows that even without a brain of their own, parasites like Plasmodium can manipulate a host's behavior by tapping into its internal hormonal gears. Another subtle strategy, interference with sensory perception, not by shutting it off, but by altering its meaning. Recall how toxoplasma in rodents doesn't block the sense of smell. The rodents still smell the cat urine. Instead, it remaps the valence of that smell from negative to positive. Similarly, some parasites affect vision or other senses in their hosts to drive behavior. There are eye flukes, trematodes like Diplostomum, that infest fish and lodge in their eyes or optic lobes. In one phase of their life cycle, they make fish behave in ways that reduce predation, perhaps blurring vision or causing the fish to stay in deeper, darker water when the parasite isn't ready. But once the parasite matures and needs the fish to be eaten by a bird, its next host, it may then cause the fish to come to the surface and swim erratically in bright daylight, virtually waving a flag to visually hunting birds. This two-phase manipulation, protect the host until I'm ready, then sacrifice the host when I am, implies a quite sophisticated modulation of the host's sensory processing and behavior over time. The parasite might achieve this by releasing different cocktails of molecules as it develops, first ones that perhaps cause slight inflammation or discomfort that drives the fish to quiet habitats, and later ones that perhaps induce agitation or affect the fish's response to light. We see that parasites aren't always jamming the sensory signal, often the signal comes through, but the host's interpretation or reaction is subverted. From ants to fish to crabs and beyond, these examples reveal parasitic manipulation as a form of multilevel neuroengineering. Some parasites act like neuromodulators, tweaking the intensity of existing signals, as toxoplasma might tweak dopamine, or as a wasp's venom tweaks octopamine levels. Others act like endocrine disruptors, shifting the host's baseline states of arousal, metabolism, or reproductive drive. Still others physically rewire circuits or destroy specific reflexes, like the dual wasp shutting down the escape reflex by targeting a cluster of neurons. The sheer range of interventions suggests that through evolutionary trial and error, parasites have explored the neural architecture of hosts in ways neuroscientists can only marvel at. And remember, many of these parasites have no neurons of their own. A single-celled toxoplasma or a fungus doesn't have a brain. The parasites are not intelligent in any cognitive sense, they have no malicious intent or conscious plan. But they are genetically endowed with neural insight. The successful manipulations are encoded in their DNA. honed by eons of selective pressure. In effect, the parasite's genome knows where to push or pull in the host's nervous system to get a specific outcome, because every mutation that improved that push or pull was favored. It's mind control as an emergent property of coevolution, not mindful scheming by the parasite. This raises a deep evolutionary question. How on earth do parasites evolve such specific and complex tools to manipulate behavior? It's one thing for a parasite to have a general effect like, make hosts sick and slow. Many pathogens do that just by feeding or releasing toxins without any adaptation for manipulation. But to manufacture a peptide that, say, mimics a specific neurotransmitter or blocks a precise ion channel in a particular part of the brain, that seems almost fantastical. There was no intelligent designer programming the jewel wasp's venom to target the roach cyscate neurons. So how did this capability arise? The answer lies in incremental evolutionary scaffolding. We can imagine the process in stages. Initially, a parasite might have a crude effect that just happened to be useful. Perhaps the ancestral jewel wasp's venom was only meant to paralyze muscles. Many wasps paralyze prey for their larvae. If one early wasp's venom happened to also induce a bit of sluggishness or disorientation in the prey, maybe by accident of chemistry, that wasp's larvae might have had a slightly higher survival rate, the prey wouldn't crawl away as far. That slight behavioral tweak, even if unintended, suddenly becomes a trait that can be refined. Generation after generation, natural selection can amplify effects that benefit the parasite. Mutations in the venom composition that made the cockroach a little less likely to wander off were rewarded by more wasp offspring. Over a very long time, this could ratchet up to the extreme precision we see now, venom targeting neurotransmitters for motivation rather than just causing global paralysis. A similar logic applies to toxoplasma. Perhaps early on it simply caused low-level brain inflammation which made rodents a bit less cautious. Inflammation in the brain often reduces anxiety behavior. If those rodents got eaten by cats more often, toxoplasma that induced more inflammation had an edge. Eventually, more refined methods, like specifically affecting dopamine or olfaction, could evolve on top of that scaffolding. Evolution often works with exaptations and co-opting existing pathways. A parasite secretes something to suppress a host's immune cell, but that molecule also affects the nervous system because immune signaling molecules and neural signaling molecules overlap. For example, some immune cytokines influence neurons and behavior, causing sickness behavior like lethargy. If a parasite secretes an immune suppressant that inadvertently makes a host less anxious, because immune-brain communication is disrupted, that parasite might gain a transmission advantage. Natural selection will then favor parasites that improve on that effect. Over many millennia, what was a side effect can become a fine-tuned primary effect. Moreover, hosts are not passive victims in this process. Host species are evolving in tandem with their manipulative parasites. This dynamic is often described as an arms race or Red Queen dynamic. Parasite and host each must continually adapt just to keep from falling behind the other. Hosts face strong pressure to resist manipulation because obviously a host that succumbs too easily may suffer reduced survival or reproduction. Behavioral manipulation by parasites is essentially another form of parasitic virulence, and hosts respond to it the way they respond to disease or predation, with defenses. One line of defense is straightforward: immune detection and destruction of the parasite before it can meddle with the nervous system. For parasites that physically enter the brain or secrete telltale proteins, the host's immune system might learn to recognize these intruders. The brain has resident immune cells called microglia which patrol for infection. Some hosts might evolve more vigilant microglia or a more robust ability to get immune cells into the brain, though the blood-brain barrier normally limits this. If microglia detect toxoplasma cysts, for example, they may release inflammatory chemicals and attempt to wall off or kill infected neurons. This could prevent the parasite from achieving its aims. Indeed, in many toxoplasma-infected rodents, one sees a certain amount of brain inflammation, possibly the result of the immune system fighting back. However, this defense has its own risks. Inflammation in the brain can itself cause behavioral changes or damage. An overly aggressive immune response might make the host disoriented or ill. Imagine a rodent so inflamed it can't function normally, easy pickings for a predator, ironically furthering the parasite's goal in some cases. So hosts face a delicate balance, an immune system strong enough to purge or neutralize the parasite, but not so forceful that it causes collateral damage that undermines the host's survival. Some species seem to achieve a sort of détente by compartmentalizing infections or tolerating a low level of parasite to avoid the costs of constant inflammation. Others might evolve an extra-tight blood-brain barrier or fewer molecular handholds for the parasite in neural tissue, for instance, altering the receptors that parasites commonly bind to. This way, even if the parasite is present, it can't easily exert its influence. Apart from classical immunity, hosts have also evolved behavioral defenses to avoid getting manipulated in the first place. These behaviors are akin to how animals avoid predators or pathogens. In fact, avoiding a parasitic manipulation is often about avoiding the parasite itself. Social animals may ostracize or avoid members of their group that exhibit signs of parasitic infection. Ants, for example, are known to carry diseased or parasitized nestmates out of the colony to prevent the spread of fungi or other parasites. In some ant species, uninfected ants can sense, through chemical cues on the cuticle, that a fellow ant is parasitized by a brain-controlling fluke or infected by a deadly fungus. They will then aggressively remove or kill that ant, essentially sacrificing one to save the rest. This is a harsh but effective collective behavior to stop the parasite's manipulative stage from kicking in. The fluke-infected ant might climb and cause others to follow or get the colony noticed by a grazer. Better to lose one ant than many. Among vertebrates, avoidance can be more subtle. Many animals learn to avoid areas or foods associated with parasites. For instance, sheep will avoid grazing in parts of a pasture where feces, and thus parasites, are concentrated. Chimps and other primates have been observed avoiding sick individuals or taking care when grooming them. Birds sometimes abandon nests that become infested with certain parasites.